Hello. Hello.
Hello.
Yeah. Are you able to hear me?
Yes.
Okay. Okay. Now let me share the screen.
Okay.
Now this problem will not come because now I got the password.
Okay.
Okay. Now are you able to see the screen again?
Yes.
Okay. So now coming to this CRISPR, right? So CRISPR is nothing but clustered, regularly interspaced, short palindromic repeats. Okay. So that means like you have some sequences which are repeating, and these sequences happen to be somewhere like 20 to 50 base pairs. Okay. So they're palindromic. You know the meaning of palindromic, right? So
Yes.
yeah. So these palindromic repeat sequences are there. In between those repeat sequences, you can see unique sequences which are not repeated. Okay. So you have repeat sequences. In between repeat sequences, you have unique spacer sequences.
Hmm.
And flanking those this CRISPR array, you have some genes, okay, which are associated with the CRISPR proteins. Okay, so CRISPR associated proteins. So, so this is the entire locus is there in many bacteria. Okay, so basically, this CRISPR array will produce the guide RNA. Okay, and those genes flanking those CRISPR array in the CRISPR locus, they will be producing the Cas9, okay, the protein, right? And
Yeah.
also there is one more RNA molecule called track RNA, transactivating CRISPR RNA. That is also coming from a region within the CRISPR locus, okay? So all these are required for the functioning of this CRISPR elements. Now, so basically what all you need for CRISPR? So you need a protein, right? So you need a protein.
Hmm.
So the protein is bound to what? Bound to a guide RNA. Okay, so you have a RNA molecule
Yeah.
which is bound to the protein. We're able to see.
Yeah.
So you can see that a significant portion of the RNA is bound to the protein, and a 20 base pair, a 20 base stretch is actually out. Okay, so this is free to interact. This 20 base pair stretch is free to interact with the target DNA. Okay,
Yeah.
so so the thing is, guide RNA is an RNA molecule. Now, if you see the guide RNA is not actually initially in the original sense, in the original bacteria. Like for example, the most widely studied bacteria is the Streptococcus pyogenes. Okay, we call them SP, SP Cas9, because initially most of the Cas9 experiments were done using yeah, SP. Okay, so Streptococcus pyogenes. So uh, this we call SP in short. Okay, so this SP Cas9 was the most widely studied. And why they chose this organism is because it has the most well-developed CRISPR system. Okay, nearly they found 120 unique spacers in the CRISPR locus of S5 genes. Okay,
Okay.
So that is why like it became a model organism, and the Cas9 from that was extensively studied. So if you see the how this guide RNA is formed, the guide RNA itself is actually made up of two elements. Okay, so you have one guide RNA. Okay, this guide RNA will
Yeah.
contain will contain two regions. It will contain the so basically the guide RNA will contain two regions. One is the CRISPR RNA, okay, and the transactivating CRISPR RNA. Okay, the CRISPR RNA itself is having two regions. One is the repeat sequence. Okay, one is the repeat, and one is the spacer. Right. So since it's actually transcribed from the CRISPR array, CRISPR array contains repeats and spaces, na? So you get at least one spacer and one repeat in your CRISPR RNA. The remaining RNA molecule, that is a track RNA, will have complementarity with the repeat region, and it will have some stem loop structures. Okay, so this complementarity is over here. So the repeat sequence of your CRISPR RNA is having complementarity with the track RNA, trans-activating CRISPR RNA. Okay, so this region. So now this is actually two parts. It's actually a RNA duplex, right? Two RNA molecules bound to one another, right?
Yeah.
This forms the guide RNA in the original sense. Now this region, okay, is bound to the protein. Okay,
Cas9.
your repeat sequence, which is attached with the track RNA, trans-activating CRISPR RNA. This acts as a scaffold, okay, to bind to the protein. It's actually the stem loop structures and the repeat sequences, they are actually adapted to bind to the CRISPR protein, Cas protein. Okay.
Yeah.
Whereas this 20 base pair stretch, which is called the spacer, that is actually the one which recognizes the target DNA. So what Jennifer Doudna and Carpentier did was, just by manipulating this sequence, right? Just by changing the 20 nucleotide stretch of this spacer sequence, one can target different genes. That means that they need not play around with the sequence which is inside the protein. Okay, so whatever guide RNA, the scaffold sequence is the one which binds the protein. That is not changed. Only the one which binds to the target DNA, the 20 base pair stretch, that is actually manipulated. You can come with based on the gene you want to target, you can reprogram that sequence. Okay, that's what Jennifer Doudna did, and that's why they got Nobel Prize. So they were able to knock out GFP gene in human cells. Okay, so that shows that like you can target any gene, okay, just by changing the 20 base pair stretch. So what was originally developed as a immune defense system in bacteria now has been repurposed into a powerful gene editing tool. Okay, now how this works we will see. Okay, how this so basically the components of CRISPR are your Cas9 protein, your guide RNA. The guide RNA itself is having two parts, the CRISPR RNA and the track RNA. Yeah, so TR, trans-activating CRISPR RNA. Okay, now going to the next slide. So as I said, this CRISPR's elements are found in many prokaryotes, but you'll be surprised that recently, just a few months before, maybe like 1 or 2 months before, they found a CRISPR-related protein, a system, CRISPR-related system in eukaryotes also, simple eukaryotes. Okay, they found something called Panzer. Have you heard about this one? It is a very recent news.
No.
Now in eukaryotes also, you have something called Panzer. Okay, it was very recently discovered, just 1 or 2 months before. Okay, And this
Okay.
Panzer is actually the CRISPR equivalent in eukaryotes.
Okay.
Eukaryotes. Now people are thinking that maybe this Panzer also can be developed into a powerful gene editing tool. And currently research is going on because it is actually for eukaryotes, it may work well for many eukaryotic cells. Okay. So, so now people are interested in Panzer, which is actually a CRISPR equivalent in eukaryotes. Okay. The original CRISPR was found only in prokaryotes. Now, so basically in this certain prokaryotic chromosomal DNA, like bacterial chromosome, you can find a locus, a region, in which you can see these repeat sequences. Okay. So you have repeats which can stretch from 20 to 40 base pairs in length. And between those repeats, you have spacers. And these spacers can be anywhere from 20 to 55 base pairs like that. Okay. So people found that, like bacteria like SP, Streptococcus pyogenes, it has got a very well developed CRISPR system, going just by the number of spacers which are there. Okay. I told the spacers are unique. So that means like every spacer uh, is unique in sequence. But if you take the repeat sequence, they're all the same, right? So when you have repeat sequences and spacers, okay, so this spacer sequences, when you do bioinformatic analysis, they found that the spacer sequences which are in between the repeat sequences, these spacer sequences are closely related with the viral genome rather than the bacterial genome. Okay, so when you try to take those spacer sequences and then you try to do some blast, you've done some bioinformatics, right?
No. As
Okay,
I
you
said,
have
I
done
haven't done
blast.
any work with the genetics.
Bioinformatics, no. Huh?
No.
You have done blast, all those things, no.
No.
Sequence, taking two sequences and comparing. Okay, so you will do, don't worry. Okay, so
Yeah.
you have a technique called blast, in which you can take one sequence and then see in the database, okay, to which genome it closely matches. Okay.
Yes.
So this way we can find out where the gene is coming from. Now, when you take these spacer sequences found in the CRISPR array and you do a alignment, okay, you basically do a blast. It closely matches with the viral genome, particularly the phage genome, bacterial phage genome. So this only led to the speculation that this this has something to do with the immune defense system. Okay, so that is how the Howard from Dew Point he came to this conclusion. Okay, because these CRISPR spacer sequences, they are similar to sequences found in the viral genome, phage genome.
Okay.
So this led to the speculation that okay, during some course of infection, this phage genome got integrated. Some portion of the phage genome got integrated, and now that actually constitutes a memory. Okay, so that this CRISPR elements are made using that, and it can defend itself against the similar infection later. Okay, so that is what actually is the purpose of the spacers. Okay, now can you see this picture?
Yes.
So you can see here you have a bacterial chromosome, and in the bacterial chromosome you have a locus region. Okay, and if you zoom into the region, you can see CRISPR array and also some genes to the other side of the CRISPR locus. Now, if you take these CRISPR array, you can see repeat sequences which are gray, shaded gray color, and in between those repeat sequences, you can see spacer sequences. Okay. And the spaces are all unique. So if you take this spacer sequence and you do bioinformatic analysis, you will find that these spacer sequences are more closely related with the bacteriophage genome rather than the bacterial genome. Okay, so this suggests that the spaces have been acquired during infection. So when the SPK9 has 120 unique spacers, it shows that it is immune to several phage infections, right?
Okay.
Several types of phages. Okay, so it is immune because it has got nearly 120 spacers. Each spacer has been acquired during one course of infection by a type of phage. Okay, so it depends on the bacteria. Some bacteria have only one or two spacers. Some bacteria have 120 spacers and more. Some bacteria don't have CRISPR element at all. Okay, so it differs. Now, if you see here, you have this Cas genes, right? So these genes only code for the Cas9, Cas12, Cas1, Cas2, these kind of genes. Okay.
Okay.
So now coming, so this is another way of looking. Okay. So you have the gene cassette, and to that side you have the CRISPR array. We can see the spacer sequences, right? And you can see that the spacer sequences are flanked by what?
Palindromic syndrome.
Huh?
What have you asked?
No. I'm asked. Just 1 second. I will rub this way. Yeah, you can see the repeat and the spacers, right? So the newly acquired spacer is shown here.
Yeah.
Yeah.
Yeah. That's on the patch.
Yeah. So what happens is, you have a small portion of the phage genome. So the phage will sit on the surface of bacteria, right? And then it will pump, like a syringe-like mechanism, it will pump the DNA inside. Okay. So this is a foreign nucleic acid, right? So when it comes inside, uh, some yet to be understood mechanism, it was shown that some small fragment, like 20 to 50 base per fragment, is cut, and then that is integrated into the CRISPR array, okay, as a new spacer. So every time a type of phage comes, a small portion of that phage genome is taken and integrated, and this is how it constitutes the immune memory for the bacteria. So every time again the same type of phage infection happens, okay, that bacteria will be able to defend itself. Okay, so this is passed from generation to generation, right? As they divide, bacteria divides now. So this is how like it's acts as the immune defense system. This is another way to look. Okay, so you can see clearly, the invading genome. A small portion of that is cut and integrated as a spacer. So here the diamond shaped, hexagon shaped things are called spacers, and the one which are shown as cubes, they are actually the repeats.
Repeats,
right? Yeah. So the repeats are all same in color because they are the same sequence, whereas the spacers are all differently colored, right?
Yeah.
Because they're all different in sequence. Now, this CRISPR array transcription happens, and you get the RNA, a long RNA is formed. We call it a pre-CRISPR RNA. Now, this pre-CRISPR RNA is subject to some processing. Okay, so in different type of bacteria, different type of enzymes are there, and they will actually be able to break down this CRISPR RNA. So kind of splicing happens. So finally, you end up with the RNA like this, which will have one spacer and one repeat. Okay, So this long CRISPR array, upon transcription, will form a long CRISPR RNA molecule, which is processed by some, some bacterial enzymes to produce this kind of repeat spacer kind of motifs. Now, this repeat spacer motif can bind with the track RNA. Okay, particularly the repeat sequence binds to the track RNA, and that forms a guide RNA. Okay, and this guide RNA is able to bind with the protein, the Cas9 protein.
Okay. Oh,
And
I
this
see.
Cas9 protein is able to target the particular region of the target gene, target genome, okay, based on the spacer sequence. Whatever sequence is there in the spacer of the guide RNA, okay. So
Yes.
the guide RNA contains a spacer part now. So that spacer part,
Yes.
whatever gene is there, so that it'll go and target that particular area in the target genome. Okay, in this case, the
Yes.
path genome. So this is how CRISPR works. Now, this is another way of putting it. So you can see that the invading DNA is there. This is the path DNA. So small region is cut from that and integrated into the CRISPR locus. You can see the newly integrated one, which is shown in red color, right?
Yeah.
And then that undergoes transcription to form the pre-CRISPR RNA. Now your track RNA is also formed from the CRISPR array, somewhere in the CRISPR array. This track RNA has some region which is complementary to the repeat sequence of your CRISPR RNA. So your CRISPR RNA has two parts, right? One, the red color one is one sequence only, but it has two parts. One is a red color region, which is the spacer region, and the black one is the repeat region, right? So this is how it gets processed. So this processed RNA molecule will be able to bind the track RNA. So complementary base pairing will happen between the repeat sequence of your CRISPR RNA and the track RNA's anti-repeat sequence. So this is able to bind the Cas9 protein. Okay. So this black region and the green region, they are adapted to bind the protein. So the Cas9 will bind. So this Cas9 protein as such is actually a protein which can undergo significant conformational change upon binding to this guide RNA. Okay, so you can go yourself and find out in the PDB. Have you heard about PDB, Protein Data Bank?
No.
Protein Data Bank, you not heard? Yeah, PDB, Protein Data Bank. You want to retrieve a protein sequence, you want to see the structure of a protein, okay, you can go to Protein Data Bank. So Protein Data Bank, this RCSB, Royal Chemical Society of Britain, PDB, it has more than 2 lakh protein structures. Okay, you can go and visualize those protein structures, because to get a mechanistic insight of how this protein works, okay, you need to know the structure, and the structure is now available. Okay, so you have the Cas9 protein alone, Cas9 protein bound to the guide RNA, and Cas9 protein with the guide RNA bound to the target DNA. Okay, all these structures are available in the PDB, and by looking at this, but the structure, one can say that there's a significant conformational changes happened going from the protein to the protein which is bound to the guide RNA. Okay, so that means the protein initially.
Hello.
And that makes this protein active. Okay, that means the
Please repeat,
nucleus
because
activity is highly regulated.
how the activation is being done?
So the activation, because this protein, okay, has a different structure. Okay, So this protein has two nucleus domains to clear the two strands of DNA. Now, what happens is if you actually uh, uh, you have two strands of DNA, so two nucleus domains are there, but this protein alone cannot cleave. Okay, it's highly regulated. So this protein has a different structure, so it will not be able to bind to the target DNA. Okay, so though it has nucleus domains, it cannot cleave. But once it binds to the guide RNA, okay,
Yeah.
a conformational change happens, and that makes it into an active shape.
Okay.
Okay, it takes up a structure which is now, which is now adapted to bind to the target DNA and cleave it. Okay, so that means like because you don't want this protein to be active in the absence of guide RNA, because then it will not be able to target. Na? See, you have so many other types of nuclease, correct? Na? Right? You have you heard about RNase?
RNA?
RNase, the enzyme which
yes.
cleaves RNA. Yeah.
Yes.
So it has no specificity, right? You can cleave any RNA, right?
Yeah.
So if you take RNase, it will cleave any RNA. Okay, whatever
Yeah.
RNA you put, it will chop it off. So that is not useful here. Okay, because your Cas9 is also kind of similarly you have DNase, right? DNase can
Yes.
cleave any DNA, right? Have you heard about
Yes.
DNase 1? Right?
Yes.
So DNase 1 can cleave any DNA, right?
Yes.
But this is actually, the Cas9 is actually will not cleave any DNA. But once it's bound to the guide RNA, then based on the sequence of the guide RNA, it will go and cleave. Okay, so that
Yes.
means like the specificity is achieved because of this binding. Okay,
Yes.
because if the guide RNA is not there, then it will never cleave. In the guide RNA it's there, then it will cleave, that too only in the area where it actually targets. The guide RNA goes and binds. Okay, so this gives a high selectivity. Okay, that's why it's a special type of Cas9, special type of nuclease. So we call it as the RNA guided endonuclease. Okay.
Okay.
RNA
I have a
guided.
question.
Yeah, please.
Where does this tracer RNA come from?
That is also coming from the CRISPR locus only. Okay. So you have some sequences which code for track RNA. Like how you have some sequences which are coding for repeat and the spacer, na? Okay. So like that, in the CRISPR locus itself, you have some sequences which are coding for the track RNA. Okay.
But when
Now,
we
yeah,
like when we like put it as a drug, like in a eukaryotic cell,
yeah,
do we like put the sequence also? Like where does it come from? Because the eukaryotic cell don't have the CRISPR locus, right?
yeah. See, actually, one can design a plasmid which will encode the Cas9 protein. Okay, and the guide RNA. So you have plasmids which can deliver the guide RNA and the Cas9 protein. Okay. So this way, and these are under mammalian promoters. Okay. So you can put a mammalian promoter and you can actually express the Cas9 in a mammalian cell. Okay. Similarly, you can put a human U6 promoter and you can express the guide RNA also. Okay.
So the tracer RNA I can also express in this way.
Which one?
The tracer RNA I can also put a sequence in the mammalian cells and it will be expressed like in a plasmid.
Yeah, we can do. Yeah. Actually, now people have simplified. I'll come to this one. People have simplified this one. Okay. They have found that, like you have this track RNA, you have this guide RNA, right? The track RNA and the CRISPR RNA, they form the guide RNA, right? Now, these two can be connected to form a single piece. So it's called now single guide RNA. Okay, so you can express the single guide RNA through a plasmid. I will tell you how. Can you see this one? This picture?
Yes.
So you can see two different RNA molecules, right? Which form the guide RNA,
Yes.
right? Suppose
Yeah.
I connect. Can you see I'm connecting?
Yes.
Suppose I'm adding some nucleotides, extra nucleotides here, and connecting these two pieces. It becomes
Okay.
one strand. Nah? Can you see? Now it becomes one
Yeah.
strand. It comes from
Yeah.
here, comes like this, comes like this. Now this is called single guide RNA. Okay. Now
Oh,
most of the guide RNA used in mammalian cells and other things are single guide RNA. Now these are engineered RNA molecules. Okay.
okay.
People found that taking two RNA molecules and expressing them under different promoters and they reconstitute inside the cell and then they go and bind to the Cas9. To in order to simplify things, they connected these two pieces. Can you see? They
Yeah.
added some 3-4 nucleotides and they were able to seal this. And they made one single RNA, which can be expressed under one promoter.
Okay.
Okay. So it becomes very simplified because this will have the same activity as the normal guide RNA. And in fact, it's easy to express because it is now one single piece of RNA, right? Can you see? Now it will become one single piece.
Yes.
So this is called single guide RNA. So this is what
Okay.
is used in most of the plasmids to express this RNA, okay, guide
Okay.
RNA. Okay, so this is how things have evolved. So you can see that the Cas9 becomes active conformer, and then this Cas9, the Cas9 as such has a domain. Can you see? It has different domains, right?
Yes. Yes.
You can see a large domain which binds the guide RNA that's called as the receptor 1 domain. It has two nucleus domains, the yellow one and the cyan color one, right?
Okay.
So these are actually the nucleus domains, and you can see this orange domain, orange color domain,
Yeah.
right? That is called as a PAM interacting domain. That is the one which binds. So the protein also binds to the protein also has binding affinity to the target DNA. Okay. Any genomic DNA, first because the protein has to first cling to the DNA. Then only the guide RNA will try to recognize the target DNA. Okay. So what it will do is, for example, if you take SP-Cas9 protein, using its PAM interacting domain, it will try to identify a unique patch in the target DNA or any DNA. Okay. So that is actually, for example, that sequence has been found to be NGG. Okay. So SP-Cas9 will recognize NGG. N could be any nucleotide followed by 2 guanines. Okay.
Yeah.
So when you have this NGG, because seeing a couple of guanines together is quite frequent in a genome, right?
Yeah.
So that means like every 10 to 12 bases you go, you can find this PAM sequence, NGG sequence, okay, to which this protein binds. But after binding to the protein, after the protein binds to the DNA, this protein will try to actually unwind the DNA locally. Okay, so local unwinding, that means like the local denaturation of the DNA will happen once the protein binds. This denaturation of DNA will allows the guide RNA's spacer sequence to form complementary base pairing with one of the denatured strands of the DNA, right?
Okay.
So this way, and suppose the spacer sequence is complementary to one of the target DNA strands, then only the cleavage will happen. So the cleavage is highly restricted. Okay,
Yes.
so it's not like any nucleus will chop, which chop it indiscriminately. So this is highly regulated. The protein has to bind to the guide RNA. Then the protein with the guide RNA has to bind to the target DNA, and then it will try to denature the target DNA locally, and then it will try to anneal the spacer sequence, the target sequence. If there is a complementary base pairing, all the 20 bases of the spacer sequence are able to anneal or able to form, able to bind by hydrogen bonds to the target DNA, okay, the protospacer sequence, then the cleavage happens. Okay.
So
So
my question.
yeah.
I have a question. Like, it's within the conformation of the Cas9 that causes the denaturation of the DNA, right?
Yeah, this Cas9 is a special protein. After binding with the, so it recognizes using this orange color domain, it goes and patches up with the some regions of the DNA. Okay, target
Yeah.
DNA. So
Yeah.
that sequence is found to be the NGG sequence. If you
Mhm.
use a different Cas9, for example, Cas9 is coming from other bacteria like
Yeah.
Francisella bacteria or Streptococcus thermophilus bacteria or Staphylococcus aureus bacteria. Okay, so
Yeah.
those Cas9s will recognize a different PAM. They will bind to the DNA using a different sequence. Okay,
Okay.
so for example, NGG, NTG is there, NAG is there. Okay,
Okay.
NNNTW is there. Like that, different types of sequences are recognized by different
Yes.
Cas9s. So to simplify things, we are talking about SP-Cas9, and SP-Cas9 recognizes the NGG sequence. Okay,
Yes.
so the protein with the guide RNA, it goes and binds first to the protein, to the target DNA, and then this using a phosphate, it has got a phosphate, you know, lock loop, okay, which actually tries to unwind or denature the DNA. Okay, this
Okay.
protein has kind of phosphate lock loops, which will
Okay.
bind to the sequence near the PAM, and then it will try to denature it. Okay,
Okay. Okay.
so so upstream of that bound region, some 20-30 base pairs will be denatured. This allows the guide RNA spacer sequence, the one which is red in color right here. This sequence will try to now anneal with the target DNA. If suppose the target DNA sequence is matching, okay, with the guide RNA, and it's bound there, then the cleavage will happen, and the cleavage will happen 3 nucleotides before the PAM sequence. But
Yes.
suppose you know, after denaturing, annealing happens, and annealing is not successful. The spacer is
Hmm.
not able to anneal properly with the target sequence. Then the cleavage will never occur. Okay.
Okay.
So what will happen is it will start sliding to the next PAM sequence. Okay. Wherever we can find another NGG, it will slide. The protein will slide and go there. Okay. So it is not going to stay in one place and keep on doing things. So it will bind to NGG. It will try to denature the sequence there. It will try to anneal the spacer sequence. If the annealing is successful, then cleavage will happen. Otherwise, it will move on to the next NGG. Understood
Okay.
now? So likewise, it's able to scan the entire genome and find complementary I mean the target sequence. Okay, whatever target sequence it matches. So that is why it's a powerful tool. Okay.
Yes.
Now let me go to the next slide. Can you see this one?
Yes.
So you are asking this question, right? This question, the picture explains. So whatever was found in nature is a natural CRISPR pathway. Now people are more interested in the engineered ones. They don't really bother about how the CRISPR-Cas9 evolved, how they got these things acquired, how it actually functions as an immune system. Okay, those actually people lost interest. Now the interest is all how we can repurpose it and use it as a editing tool to target human cells and other cells. Okay, because for gene therapy you need to target the human cells, na? So
Hmm.
more research nowadays is concentrated, more funding and more research is actually to develop CRISPR as a gene therapy rather than study the natural evolution of CRISPR in the bacteria. Okay. So
Okay.
still, some researchers are doing this one. But anyway, the story is clear, right? So whatever originated in the as a immune system in the bacteria has now been successfully repurposed into a gene editing tool. Now you can see here comparison. Okay. So here you can see two pieces of RNA, right? The track RNA and the CRISPR RNA, which forms the guide RNA, right?
Yeah.
Now here these two pieces have been connected, right? Can you see here? This is a single guide RNA.
Yeah.
So the single guide RNA also binds the same way to the Cas9, okay, and you'll be able to. So the advantage is this entire RNA molecule can be expressed under one promoter. Can you see the red
Yeah.
part and green part? So the blue color one is the promoter, and this guide RNA is cloned. The DNA sequence of the guide RNA is cloned here. So this will express inside the cell. Okay, once you put the plasmid inside the cell, it will express into a guide RNA. The same plasmid contains another promoter which will express the Cas9 also. So under one promoter, you have Cas9 as well as the guide RNA. So if you take this plasmid and put inside a mammalian cell, like a human cell, it will finally encode the Cas9 protein. It will also encode the guide RNA, and in situ, inside the cell, they will recombine and form a complex, which will become the active Cas9 complex. Now this complex will be able to enter the nucleus because that is where the DNA is found, right? So you will have a nucleotide location signal also, NLS, a small peptide attached to the Cas9, because Cas9 is a bacterial protein. Now, if it has to go inside the eukaryotic nucleus, it needs a sequence, like a permit.
Yes. Yeah.
So they attach a NLS sequence, nuclear replication sequence. Okay, so this sequence allows the it has to go inside the nucleus and nucleolus, and then only can find the DNA, right? The target DNA, right? So the Cas9 is now always having an NLS sequence attached. So the plasmid itself, you will code for NLS, Cas9 NLS. Okay, so that this Cas9 will be able to enter the eukaryotic cell nucleus where it can find the DNA, and then it will be able to edit.
Okay. So the nuclear localizing sequence, I will be adding it at the very end, right?
Yeah, all the plasmids nowadays, all the plasmids by default they will have. All the Cas9 expressing plasmids will have the NLS signal. It can be in the N terminal, it can be C terminal, either way. But I think people prefer the C terminal. Okay. So you have the NLS signal, which will allow the protein to go inside, the protein guide RNA complex to go inside, and then where the real business happens, the cleavage happens.
Okay.
Now, people are finding that instead of using plasmid, one can go with the mRNA also. Nowadays, mRNA in vitro transcription technology is developed, right?
Yes.
Yes or no? People are making mRNA vaccines, people are making mRNA therapeutics. So mRNA of Cas9 also is available. Okay. So many companies now sell Cas9 mRNA. The advantage of getting Cas9 mRNA is, Cas9 mRNA will be, Cas9 mRNA will be able to, it will be able to express into protein much easier. Okay, because when you put out a plasmid, sometimes the promoter incompatibility will be there. Sometimes the expression is not guaranteed. Going from see, even though it's a human cell, in different cells the expression will be different.
Yes.
Okay, so there are so many types of human cell lines, right? So in each cell line, the plasmid expression is not uniform. So sometimes you don't, the Cas9 is not formed properly. Okay, enough Cas9. So the stoichiometry of the Cas9 guide RNA is not maintained there. So sometimes you have issues when you do plasmid-based expression of Cas9. There are some issues. Sometimes you don't get enough concentration. Sometimes what happens is the plasmid keeps on expressing the Cas9, that even after the editing has happened, still the Cas9 is expressed, and that will lead to off-target editing. Okay, so these are issues of the plasmid. So when you use the mRNA immediately we will because whatever mRNA is there, it'll become protein, right? mRNA will become protein. So there the promoter, nothing is there, right? So it's easy. The mRNA can go to the ribosome, it will get made into the protein. the protein with the NLS sequence will go inside the nucleus and do the editing along with the
Yes.
guide RNA. So people are trying to co-deliver the mRNA and the guide RNA using lipid nanoparticles and other systems. Okay,
Yes.
inside the human cells. So once they're inside, because the mRNA expression into protein is very straightforward. Okay, so one can quantitate how much mRNA you need for delivering this amount of dose, this amount of Cas9 protein. Okay, but in the plasmid, the expression levels keep on changing. That's the problem. So people have now adopted Cas9 mRNA, and one can also form the protein. For example, you can in the bacteria itself, you can express Cas9, purify the Cas9 protein, and you can using in vitro transcription, you can make the guide RNA outside. Okay, so that guide RNA and the Cas9 protein can be combined, and this ribonuclear protein, okay, can be delivered inside, right? So this is also
Yeah.
possible. So now this is also highly preferred because the thing is, you take the protein, you take the guide RNA by in vitro transcription, you form the guide RNA, and the guide RNA and the Cas9 are assembled, and then they are sent inside. So this way they are very quick acting because the plasmid has to go inside, it has to multiply, then it has to replicate, and then it has to express. It takes more than 72 hours for the plasmid to work. But when you give us a mRNA or when you give us a protein, Cas9 protein, it's almost instantaneous. The editing will happen immediately. Okay, and after the edits are made, the protein will be removed from the system, right? Yes or no? The protein half
Yes.
life is around 70 to 80 hours only, right? Any protein you inject
Yeah.
inside will not be there forever. So once the business is achieved, it will go, the Cas9 will vanish, right?
Yes.
But when
Yes.
you use a plasmid, it takes 3 days after 3 days only it start expressing. Sometimes it's slower, okay? And then it will linger in the cell for a long time, which is unwanted. Because after
Yeah.
editing is done, why do you need the Cas9 inside the cell, right? So these are the so people are nowadays not using plasmid, mostly for gene therapy and all. They tend to use mRNA or the ribonuclear protein. Okay,
Okay.
so these are the things. But anyway, like gene delivery is another important thing, and therefore people use viral based delivery also. Okay, the
Yeah.
Cas9 and the guide RNA are packaged into um, viral compatible vectors. Okay, and like some plasmids which are lentiviral plasmids or uh, AAV. AAV means adeno associated virus. Okay, so these are preferred in gene therapy, but nowadays the trend is changing. Okay, so people are using AAV based gene therapy, right? Not only for CRISPR, for many other things. Now you can package the Cas9 into AAV also, right? But the thing is that the AAV, its load, now it can carry the nucleic acid load is quite
Yes.
small. A Cas9 being a large protein, its gene is quite long, right? So
Yeah.
people find it difficult to express Cas9 through viral delivery. So
Okay.
they break down into two parts and assemble, and then they make sure they get assembled inside the cell. So these are all done, but nowadays people are preferring smaller Cas9s. So whose gene will be smaller and it can easily get packaged into AAV virus. Okay.
Yes.
So now I will give a break for 10 minutes.
Okay.
Okay, is it okay?
Yes.
So I'll give a break for 10 minutes. Then we can resume after 10 minutes.
Okay.
Meanwhile, any questions you have, you can ask.
Okay.
Hello, can we resume? Hello,
Yes. Yes. Can you hear me?
can we start?
Yes.
Yeah, are you able to see slide?
Yes. I can see the slide.
Yeah, so here you can see like this is the genomic locus, and this is where you target sequence, right? So when I'm designing my guide RNA, I have to first look at the sequence, which where I'm going to target. Okay, so I have to identify a unique sequence in my target gene. Suppose
Okay.
I want to knock out a um, um, uh, there is one gene, like, is there any breast cancer causing gene is there?
Where?
Do you know any gene which causes breast cancer? Oncogene.
BRCA?
Yeah, okay.
BRCA?
So suppose you want to knock out BRCA. Okay.
Yeah.
So what do you do? You have to see a unique stretch of sequence in the BRCA gene, which is unique to BRCA, which is not found in other genes. Okay, because see, you have only 4 types of bases, na? Right? The entire
Yes.
genome is made up of only 4 types of bases. So it is
Yes.
always possible that similar sequences are found in many genes, right? Some stretch of, if we suppose randomly take a stretch of DNA, for example, a 5 to 6 base pair, that the 5 to 6 base pair stretch will be found in at multiple sites in the genome. For example, ATCCCG. This ATCCG can be found in multiple places, right?
Yeah.
But when we go for a longer sequence, like a 20 base pair stretch, right? Then you know that same 20 base pair stretch of you may not find everywhere, right? Rarely you will find it, right? But when you use bioinformatic tools and find in that gene, in that BRCA gene, okay, BRCA gene, which sequence is unique to that gene, which is not found elsewhere. Like I don't want any other gene to have that sequence that we're going to select. Okay.
Okay.
So I'm selecting one unique sequence within the BRCA gene, which is actually not found or not similar sequences is not found elsewhere. Understood? Now, in other parts of the chromosome. So that means when I'm targeting that gene, that area, only that gene will be knocked off, right? The other genes will not undergo any kind of change. So to impart specificity, because your Cas9 is a cleaving enzyme, now it cleaves your DNA, right? So you have to be very careful. So you have to select using bioinformatic tools. You have to select a unique stretch, okay, where you want to target, where you want the guide RNA to cleave, okay. So you select the target. So you have a gene, you decided to knock out the BRCA gene. But within that gene, which area you want to target? The 20 base pair stretch you have to select.
Yes.
So after selecting that sequence will be grafted into your guide RNA. Right? So the 20 base pair stretch in your guide RNA spacer sequence is decided by the target sequence. Understood? So whatever this region is there, na? The target gene. That sequence only is actually taken and you are actually basically programming the RNA, right? So this sequence. So if you want to target some other gene, okay, you want to target some other, for example, S100A4 gene, then again this 20 base pair stretch only will change. For that gene, I find one unique sequence where I want to target. That sequence I grafted here. Okay, so it is as simple as that. Okay, the remaining part of the guide RNA is not changed at all. It is the same. Whatever. So you can see the guide RNA is here. Can you see?
Yeah.
The guide RNA is over here. It starts here. Can you see the blue color one?
Yes.
So this is the 5' end of the guide RNA. It goes all the way. Then it actually becomes red in color. Can you see?
Yes, that's tracer RNA.
Yeah, that is actually the scaffold sequence, the repeat and the track RNA, right? So now they are
Okay.
all fused into one. Can you see?
Yes.
Now one single piece of RNA, right? So this is the business end of the RNA molecule, guide RNA molecule. The blue one, what is shown in blue color, na? This is the business end because this is the one which binds to target DNA, right? So this spacer sequence have been zoomed and shown here. So basically, first the yellow color one is the protein, right? So the protein will recognize the PAM sequence, the TGG. I told the NGG, N to N nucleotide. So it recognizes the NGG sequence and binds there. After binding, it will denature the sequence upstream of the PAM. Can you see? The hydrogen bonds are all missing here. Can you see? Because it is denatured, right? Denaturation means what? The hydrogen bonds are all broken, right?
Yes.
So the hydrogen bonds which are connecting the two strands, so locally it becomes denatured. Okay, because of the activity of the protein. The protein uses a phosphate lock loop to unwind the DNA.
Okay.
And so this allows the guide RNA spacer sequence to establish complementary base pairing with the one of the strands of the target DNA. So once this is perfect, all the 20 bases are able to anneal perfectly. Okay, then the cleavage happens. So this is a very restricted cleavage. It doesn't happen all the time. Only when the 20 bases of your guide RNA is able to form complementary base pair in the target DNA, then only the cleavage happens. And the cleavage happens 3 residues before the PAM sequence. Okay, PAM is this NGG, TGG here, right? That is the PAM sequence, which the protein binds. And it cleaves 3 nucleotides before that in both the strands. Can you see? The arrow marks, right? Red color arrow marks. These are the cleavage sites where the cleavage happens. Okay, the protein will cleave there. So this is actually a um, very perfect model for specific cleavage, right? Were you able to understand this one? Any doubts here?
No.
Can I move to next slide?
Yes.
Yeah. So you can see here, this is the original structure, crystallographic structure. So these are all just models for schematic illustration. This is actually the experimental structure solved by crystallography, right? So NMR and crystallography are very powerful tools in getting the structure of the protein. So this is the crystal-based structure, crystallography, X-ray crystallography-based structure. You can see that the guide RNA is here. The target complementary region is shown here in the red color. So this is one single piece of RNA, guide RNA, and you can see the protein. Okay, so the protein is shown now. A lot of helices are there, right? So you can see that different domains of the protein have different color, just for understanding. Okay, and you can see this orange color one, which is the PAM interacting domain. The blue one and the yellow one are the nucleus domains, right? And this long one, this top domain, that is the receptor domain, which binds the guide RNA. Okay, so after the bind the guide RNA, you can see that a significant conformational change. Can you see? This yellow domain has come down and the blue domain has moved up, right? The receptor domain here has moved up here. Can you see?
Yeah.
You compare this structure with this structure, a lot of changes there. This is brought about by guide RNA binding. When the protein tries to bind the guide RNA, it undergoes a significant conformational change. Domain rearrangement happens. Okay, so this protein folds into different domains which have different function, and these domains rearrange spatially, okay, to give an active conformation, which is ready to bind and attack the target DNA. So these structures are there in PDB. We can go to PDB. Can you see this one?
Yeah.
The Cas9 alone. So you can, I can show you how to use the PDB. It's a very important bioinformatic tool. Just force CMP and force Z2 on force Z2. So I'll stop share and I'll show you the PDB. Fine.
Okay.
So PDB we go. Okay, shall I share the screen? Are you able to see the screen?
Yes.
Yeah, so when you type PDB in Google, this RCSB PDB, Royal Chemical Society of Britain PDB is coming. Okay, so if I go there and I type this sequence, the PDB code 4CMP, 4CMP. So this is actually the every structure deposited in PDB has a unique ID. Okay, so this 4CMP is actually the one with only the protein, no guide RNA. Okay, so this is the Cas9 protein without the guide RNA. It has a different structure.
Mm.
Can you see this one?
Yeah.
So you can go here and you can view the structure in detail. Because you may get better insight, mechanistic insight when you have the structure. Can you see the structure?
Yes.
You can rotate and see. Okay. So this is the Cas9 protein without the guide RNA. Okay. Now what people have done, they added guide RNA and again crystallized it and got the crystallized structure. They found that after binding guide RNA, there's a very huge conformational changes happened. Okay, so these are all revealed by extrographic structure studies. Okay, so let me show that structure also. So I type 4ZTO, the same Cas9 protein, SP-Cas9, but now bound with the guide RNA. No, this is different. 4ZTO. Once again. 4ZTO. Okay. So this O is not 4ZTO, 4ZT0. 4ZT0. 0. The PDB codes are very similar. Can you see? And this is the Cas9. Can you see streptococcus pyogenes Cas9 complex with the single guide RNA at 2.9 Armstrong resolution? So if you see here, can you see this one?
Yeah.
You can see the protein and the guide RNA. Okay. Can you see the guide RNA in violet color, right?
Yeah.
The violet color one is the guide RNA. This is an experimental structure, two structure. Okay. This is not a model. It's the original structure. This is how it originally looks like. So this is got by a technique called extrography. Okay. So you can see the protein bound to the guide RNA, and the protein has changed structure while binding the guide RNA. Now it is actually in attacking conformation. You can now readily bind the DNA, target DNA, and it can actually, once the guide RNA is able to form complementary base pairing with the target strand, it will then start cleaving. So it has to position its nucleus domains such that they are going to attack the two strands of the DNA. Okay. So till the annealing takes place, it will wait. So it's very it has got a lot of restraint. Okay, it's not just like that randomly clean. But even though with so many restraints, if the target is not selected properly, if suppose I am selecting 120 base pair stretch, okay, where I'm going to clean. But what happens is that 20 base pair stretch has some kind of similar sequences available in another part of the chromosome. Okay, maybe 19 or 18 are matching. Okay, suppose 18 bases are matching. Okay, so 1 or 2 are different. So there's a high chance that this Cas9 will go to that part of the chromosome and cleave that gene also. It's possible.
Yes.
Okay, because only a few base pairs are different from the target sequence. Okay,
Mhm.
so what your intent to cleave, that is the target sequence, and
Yes.
what you end up unintentionally cleaving is the off target. Okay,
Yes.
so off target means some sequence which is similar to the target sequence, except for a few bases. So we call them as mismatches. Okay,
Yes.
so one or two bases of mismatch. So that is the off target. So it's very similar in sequence. So that also can be potentially cleaved. So this has to be taken into account. That is why we need a bioinformatic tools to actually come out with a unique stretch of sequence which has no off targets.
Hmm.
Okay. Now coming to that bioinformatic tools. So you have also structure with the DNA also. So Cas9 DNA, target DNA, that is also available. Cas9 guide RNA and can you see this one?
Hmm.
You have all three here. You have the Cas9, you have the DNA, and you have the RNA.
Hmm.
It's an attacking conformation. Can you see this one?
Yes.
So you can see that your protein is there. The violet color one is the guide RNA, right?
Hmm.
And you have the DNA also, double stranded DNA. You see? Double stranded
Okay.
DNA also there. So now you can see the DNA and RNA are paired also, right?
Yeah.
Can you see this pairing here? Can you see
Yes. Yes.
this pairing? So this is the non-target DNA strand. This is the
Oh, okay. Non-targeted region.
target proximal strand. This is the guide RNA. So the green one is the guide RNA here. The green one is the
Yes.
guide RNA. A small portion of DNA is shown. Okay.
Hmm.
So like that, these are all experimentally determined. So this only gave mechanistic insight into how the protein functions. Okay. Now you have so many bioinformatic tools. So I can show you a couple of tools. For example, you have a tool called from Harvard University. Harvard University, you have a tool called Chop Chop.
Okay.
Okay. So are you able to see screen?
Yes.
Hello.
Yes.
Uh, yes, just 1 second. One delivery has come. Just receive the parcel. Just 1 second. Where, sir? Where are you? Huh? So this chop chop, you can also try around with me.
Okay.
Okay. So I will tell you the rules of designing the guide RNA, but generally before that I will just expose you to some tools. So how these tools work, I will explain to you. Okay. So can you see here? This is from Harvard University. It's one of the most popular tools for designing the guide RNA. Okay.
Okay.
So for first designing the guide RNA, what you do? You design you have to come with the target sequence, right? What gene you want to target? What question you want to address? For example, every gene therapy, there's one gene you want to target, right? Sickle cell anemia means you want to target the hemoglobin beta gene, right? Yes or no? If you want to target muscular dystrophy, Duchenne muscular dystrophy, then you have to target the dystrophin gene. Okay, so you decide what gene you want to target. Then in that gene, generally eukaryotic genes, right? They have multiple codons. Yes or no?
Yes.
I mean multiple exons.
Yes.
So it's not like in bacteria. Bacteria has no intron exon, only the coding sequence will be there, right? But in a eukaryotic gene, like particularly human genes, you can see like significant 90% and above, 99%, close to 98 to 99% is actually non-coding, right? So you will see long stretches of non-coding sequence and in between small islands of coding sequence, right? Now,
Yes.
some gene, for example, you take the DMD gene, muscular dystrophy gene, it contains nearly 79 exons. Okay, so you have to select which exon you want to target, right? So that is very important. Suppose your purpose is to knock out, okay, which gene you want to be, which exon you want to target. If your purpose is not knockout, because you don't want to knock out hemoglobin B gene and dystrophin gene, no, you don't want to knock out, you want to repair it, right? It's faulty, you want to repair it. So for repair, you have another mechanism, knock-in mechanism. Okay, you want to knock in and swap some correct sequence with the wrong sequence. Okay, knockout means deleting a gene. Suppose you have onco gene which is causing cancer or HIV kind of a CCR5 gene. Okay, so you want to knock out the gene which is not useful for you, then you can go for knockout. But in case you want to repair a gene, you cannot do knockout there. You have to specifically edit that sequence, right? So that is what it becomes more interesting. Gene therapy, most of the time, it is actually repairing a gene, right? It's not about knocking out a gene. Okay. Sometimes you do knock out of a gene, but many other times you repair a gene. For example, sickle cell is there, right? So that's sickle cell anemia is because of one single mutation in the hemoglobin B gene, right? So that particular gene, that particular nucleotide only you have to target, right? It's very specific. The mutation point is the pathogenic mutation is very specific, and you want to revert only that mutation, right? So that means like you need more. You need a lot of other things also to target the particular gene, a particular place. Okay, specific editing is more difficult also. Okay, compared to knockout. So if you want to knock out a cell, it's easy. Okay, people are doing a lot of knockouts in cell lines to study. For example, people are interested in developmental biology, embryo, so they want to knock out some gene and see how the cell behaves. Okay, they take embryonic stem cells and then they knock out some gene and then they see how the development is affected. Okay, for functional validation of genes, CRISPR can be used in cell lines. Okay, but when it comes to gene therapy, this knockout, we should be very careful. So generally, people try to repair the gene, right? So this tool offers you all those things. You can do knockout, you can do knock-in, you can selectively activate a gene also, and you can selectively suppress a gene. Okay, so nowadays there is no need to cut or cleave a gene, because cutting or cleaving a gene, you will not get FDA approval for therapy for human use. Okay, FDA is very strict about use of Cas9. So you will so any treatment that is done using Cas9, you have to get a FDA approval, right? And FDA approval is generally granted for smoother variants of Cas9 or milder variants of Cas9, like the base editing and the prime editing. Okay, so these techniques they don't cleave the two strands. They either cleave one strand or they don't cleave at all. They only modify the sequence. Okay, for example, in sickle cell anemia, the A to T transition, I just want to change the nucleotide. I don't want to cleave the DNA because that way it's more safer, right? If I'm able to selectively modify one sequence, for example, some SNP, some single nucleotide polymorphism is causing a disease. I want to revert that mutation without cleaving the DNA. That is now possible with base editing and other novel methods of CRISPR. Okay, the conventional Cas9 is not used in gene therapy. The modified variants like prime editing, base editing, those are the ones which are successful in gene therapy. They are the ones which are getting approval. Okay, so we will concentrate initially to understand how Cas9 works. We will go through this one. Then later we will go and visit those which are used in clinical settings, right?
Okay.
For example, the base editors and the prime editors which are now used, which have got FDA approval. Okay.
Okay.
So we will visit that one. Okay. For example, in CAR T-cell therapy also, people use those kind of things only, the base editors. Okay. So because people are afraid of double stranded cleavage, because when you cleave both the strands of DNA, they're pulled apart, na? Right? Suppose you have a tight rope, you cut the rope, what will happen? The rope will fall apart, right? Right? Putting them back together is not so easy, right? Yes or no?
Putting them
Hello?
back?
Huh?
What did you say?
Suppose you have tight rope, ROPE, okay?
Yes.
And you cut the rope, what will happen? The two ends will fly apart, na?
Yes.
So putting them back together is not so easy, right?
Yes.
So if we have chromosomal DNA and then you cut the DNA, both the strands are cut. That means they fly apart, and putting them together is very difficult. So that is why double-stranded cleavage is not encouraged in gene therapy. Okay, but if we have only one strand cut, still they are holding together, right?
Okay.
Right. So so no big deletion of chromosome happens there. Okay.
Okay.
So exonuclease will not come and cleave there. So because only one strand is cut, and then you do the repair, and then the nucleotide is modified. So this way, it offers a safe way to edit, right? So we'll visit those things also. Okay. So now you can target any, not only humans. This software allows you to target any kind of organism, bacteria, fungus, animals, mammals, plants, all these things. Okay. And you can also give the gene name or the gene coordinates, or even you can paste the target, paste the gene also. We don't, you want a selective portion of the gene, the long gene is there, only a selective portion, you can paste the gene also. Okay. And you can use several variants of Cas9. Cas9 nickase. Cas9 nickase will only nick the DNA, it will not cleave the DNA. Nick means only single stranded cleavage. CPF1, this is a novel Cas9 with a smaller variant of Cas9. Okay, CPF1. Your Cas13, which will cleave the RNA. Okay, and also like the old talents. So this software supports many such things. You can also go and select the different, whether you want to target the coding sequence, whether you want to target the exons, all those things. And Cas9 also, what type of Cas9? If you want to use SP Cas9, then NGG is the PAM. If you want to use some other Cas9, their PAM sequence should be identified and then selected. And anything not here also, custom PAM also supports. You can type the novel Cas9, very new Cas9s are coming now from different bacteria. So they have different PAM. Those PAM sequence can be typed here. Okay. So this is a very powerful tool. So today we will end up here. Okay.
Okay.
Because it's 2 hours daily, na? Right?
Yes.
So we'll end up here today. You need some time to read also. Okay. So this video will be posted to you. Okay.
Okay.
And you can go through. And from tomorrow we'll start using the software. We will design knockout. We'll design knock-in, repair.
Okay.
Okay. We will take some use cases like sickle cell anemia, thalassemia, muscular dystrophy, and in those genes how to do the editing. I will explain to you. Okay, using bioinformatic tools. Okay.
Thank you so much.
Okay.
Yeah.
Thanks. Yeah. Bye.
Yeah. See you tomorrow.
See you.